Hello, my name is Ruslan Dmitriev. I'm a professor at the Tissue Engineering and Biomaterials Imaging Group, Ghent University. The topic of my talk is Fluorescence Lifetime Imaging Microscopy, FLIM, normally pronounced as simply FLIM, and of course, organoids. So, what is FLIM? As you can probably know, fluorescent compounds used in microscopy are illuminated and emit the light. What we normally observe is the intensity of their fluorescence within certain regions of the electromagnetic spectrum. However, between the events of light absorption and emission, every fluorescent compound spends some time. This time is called fluorescence lifetime, and it can last up to nanoseconds. Amazing thing is that if you can measure fluorescence lifetime, you can probe a multitude of biochemical processes. For instance, interactions of the fluorescent compound with various quenchers, or even probe intermolecular interactions using Foerster resonance energy transfer phenomena, or simply FRET. To give you an example, you can see a microscope image of the dye staining mitochondria, shown as yellow. However, if we look at these from the point of view of fluorescence lifetime, here the color will indicate different values. We can start then gain more information on the mitochondrial state and their heterogeneity. In addition to just probing mitochondrial state, FLIM can be used in many other microscopy-based experiments to probe, for example, pH, lipids, or mechanical forces experienced by the cells. Striking example of the use of FLIM is monitoring of cellular autofluorescence, or so-called optical metabolic imaging FLIM. Typically, all the live cells exhibit blue autofluorescence, which is considered to be sort of junk signal, interfering with the, for example, antibody staining. However, if you expose live cells to the two-photon excited FLIM microscope, you can observe that cells will exhibit different fluorescence lifetimes. In the presented case, cell type 1 has longer fluorescent lifetime, which indicates a higher reliance on the oxidative phosphorylation and higher activity of mitochondria. In contrast, cell type 2 will exhibit shorter lifetimes, which will tell you that the autofluorescent pigment NADH is mainly involved in glycolysis in these particular cells. Indeed, such microscopy method can be combined with the use of transgenic reporters, such as LGR5-GFP, and this would help segmenting regions with different metabolism of the cells. Therefore, by simply measuring cell autofluorescence in a label-free setting, one can study mitochondrial function and cell metabolism. And using this method, it was recently found that measured cell metabolism strongly depends on the cell composition and the growth medium in which you can measure. So now I would like to introduce you to the organoids. Organoids represent three-dimensional cell aggregates which normally are derived from stem cells and display cell composition and functional features of the tissues and organs that they mimic or reconstruct. For instance, small intestine organoids exhibit cell composition of the native gut epithelium. These in vitro models have potential to rebuild the tissues using, for example, technology of induced pluripotent stem cells or iPSCs. In addition, organoids can be produced not only from the gut, but also from other tissues such as retina or the brain. Which problems can we address with organoids methodology? For cancer, we can produce patient-specific organoids and develop better drug therapies. We can also gain deeper insight into stem cell biology using organoids. We can address etiology of inflammatory bowel disease, and for brain, we can gain deeper insight into neurodevelopmental disorders. Let me tell you here a little bit about our work on neural organoids. There is a gene called FOXG1, and its insufficiency can be linked to the development of autism spectrum disorders. Normally, it's very difficult to study what happens in the brain of the patient, but with organoids we can get iPSC, or induced pluripotent stem cells, produce neural organoids, and start to learn what is happening during the early development of the brain in this particular individual. And neural organoids are quite big, but still compatible with fluorescence lifetime imaging microscopy. We can label them with the functional dyes, informing on the brain development, and look deeply into the organoid to see how FOXG1 insufficiency affects neural development. So you can see from this video that We can successfully analyze large live samples with depth of up to 100 micrometers and a single cell resolution. With FLIM, we can also see the functional heterogeneity in the developing neural tissue. To conclude my talk, I'd like to remind you that FLIM enables for live multi-parameter imaging of cell function in 3D. We can study not only neurodevelopmental disorders, but also cancer, or, very interesting for the future and the sustainability, we can use FLIM to understand how nanoplastic pollution can influence the cells in our guts. I would like to thank my team, collaborators, and funders, and thank you for watching.